Hello dear students. So in this session we are going to discuss about methods of analysis, that is chapter 4.2 under unit 4. So in our previous class we completed with chapter 4.1, that is chromatographic techniques. So we discussed about thin layer chromatography, paper chromatography, column chromatography and gas chromatography, their principles involved in that techniques, and also what are the advantages of particular chromatographic technique, and also we saw few examples of that. Okay, so that completed chapter 4.1. So in today's session, so we will be having a brief introduction on methods of analysis, and also what are the different types of analysis which are used, that too in chemistry, that is what we call as chemical analysis. What is meant by chemical analysis, what are the different two types found there, and what are the different techniques we are going to use to do those analysis. So all this will be understood in this session. So before moving on to the part, let us have a brief introduction, like what is meant by chemistry. Okay, so chemistry is a branch of science which deals with the study of chemical properties, composition, bonding, and structure, manufacture of various compounds. So all this, the study of all these properties is totally considered as a branch of science which is called as chemistry, right? So we will see what is meant by all these things. So before that, what is the actual meaning of analysis? So why we have to do analysis? So the actual English meaning of analysis is the careful examination of the different parts or details of something. Okay, so in Kannada it is visleshane. So Visleshane is the exact Kannada term which is meant by analysis. Okay, so this analysis is everywhere in our day-to-day life. We do analysis in every aspect. So in while doing shopping also, while purchasing or new dress or new materials or new things, so we are going to analyze it. So how far it is very good, how far it is usable, how economical it is. Okay, so whether it is cost effective or not, we are going to analyze it. Okay, in the same way while buying the food products, so whether it is healthy or not, whether it will improve the proteins, or whether it will give enough sufficient energy for our body. So everything we do analysis. In the same way we also do analysis of students in class. Okay, a teacher will analyze a student. So the different methods or steps which he uses to analyze a student is, so assignments will be given and he will check how efficiently the student is going to submit the assignment, and also the internal assessments what are conducted. By seeing that, so we will analyze a student. In the same way, so we have to analyze the chemical compound which is synthesized or which is extracted, so that has to be analyzed. Okay, so for that purpose, so we use these techniques, different techniques, to study the chemical properties means, so how it is going to react with the each other, whether it is having a acidic property or whether it is having a basic property, and what are the compositions means, what and all the elements are present in it, what and all the ions are present in it, so that will be studied. And also bonding, how the chemical bond has been formed. Okay, so how the interaction between the elements are observed, and also the structure, whether it has got a pyramidal structure or tetragonal structure, or how does the structure appears to be. So that will be understood with the help of analyzing a compound, and also how we have manufactured of various compounds. So all these will be studied with the help of methods of analysis. So that is what we are going to discuss in this chapter, that is 4.2. And now, what is meant by chemical analysis? Chemical analysis means determination of composition of a chemical compound is known as chemical analysis. That means what and all the compositions are present in it, so that is determined by subjecting that particular compound into the examination. We are going to find out what are the compositions of that particular compound. That term is called as chemical analysis. So this chemical analysis is classified into two types, that is, one is qualitative analysis and another one is quantitative analysis. Okay, so here if we observe the name, that is qualitative and quantitative, so it gives us what exactly this analysis means about. Okay, so a qualitative analysis is nothing but, so it is a branch of analytical chemistry, so which deals with the identification of elements or ions or molecules of a sample material. That means quality means what? Quality means what is present in it. We are not concerned with how much amount is present, we are concerned with only the quality. So what element is present, so what ion is present, or how the molecules are present, so that is what is called as qualitative. Here we are not looking for the amount, we are just concentrated on what is present, what different types of elements is present. So that is why identification of elements or ions is called as qualitative analysis. So it is done by subjecting the sample of a material to a series of chemical reactions systematically. Okay, so how we will do the qualitative analysis is, so you can just imagine, or you can just go back to your PUC classes where you performed a semi-micro qualitative analysis of given inorganic salt mixture, right? You had given a salt mixture and you were asked to find out what acid radical is present and what basic radical is present in that given inorganic salt, right? So that is nothing but an example of qualitative analysis. So here sample or a sample material is subjected to a series of chemical reactions. We are going to perform a series of chemical reactions, and the identification of the constituents is done on the basis of qualitative observation or the chemical reaction. So the systematic qualitative analysis is governed by principles like solubility in a solvent, common ion effect, solubility product, etc. Okay, so these are the main principles which are involved or governed in a systematic qualitative analysis. That is, one is solubility in a solvent and common ion effect and solubility product. Okay, so now what are the examples of qualitative analysis? The first example is, as I told, a systematic qualitative analysis of inorganic salt, which experiment we have already performed in your second PUC classes, that is identification of acid radical and basic radical, is one of the example of qualitative analysis. In the same way, we can also do systematic qualitative analysis of organic compounds also. In the same way how you have performed in an inorganic salt mixture, in the same way we can also do the qualitative analysis for organic compounds, and also qualitative analysis of ores, alloys, etc. So these are the three different examples which we can give for a qualitative analysis. Further, so what we have to see is classification of qualitative analysis depending upon the size of the sample available and the techniques. Okay, so this is the main thing. So we are going to classify this qualitative analysis into based on the two parameters, that is, one is sample size. So depending upon the sample size we are going to do the classification, and the next classification will be depending upon the techniques what we are going to use. Okay, so first we will see depending on the size of the sample available for the analysis. So we are classifying it into four types. The first one is macro analysis. Okay, so what is meant by macro analysis? When we have a large quantity of sample, okay, so we need not to worry about oh, I have only 1 gram of compound, or I have only 0.5 mg of the compound, so I should be very careful in using that. So you need not to worry like that. Okay, so you have got large quantity of samples and reagents are available for the analysis. So then we are going to employ macro analysis. So here the reagents or a sample which is used, so that will be used in a gram scale. And okay, so we are going to use it in a gram scale. So these macro analysis experiment is carried out using gram scale. So for example, analysis of soil. And okay, so analysis of soil or a systematic analysis of inorganic salts. When we have large quantity of samples and reagents, we can do the systematic analysis of inorganic salts in macro analysis method also. Okay, so for example, first example is analysis of soil. We need not to worry about the sample of soil. Okay, soil is everywhere. We have enormous amount of soil, and we can take how much ever we want, and here we don't have any scarcity of sample or a reagent. So that is why so here we can use 1 gram or 0.5 grams or even 5 gram 10 gram of soil sample also can be used. So that is why this is classified under macro analysis. Okay, and the next thing what we have to notice here is, in this method, so no special apparatus or no special skills are required. Okay, so this method requires more time, and since we are using large amount of sample and reagents, definitely it requires more time. So this is all about macro analysis. And next, moving on to the second one, that is semi micro analysis. So when we do semi micro analysis, that is when small quantity of sample and reagents are required for available of analysis. When we have only small quantity of sample and reagents, so then we will do it in a centigram scale. And okay, so the semi micro analysis is carried out using centigram scale, and this method is economical, and also this is a quick method, and in this method special apparatus and skills are required. And for example, systematic semi micro qualitative analysis of inorganic salts. And okay, the same example, so there means the special apparatus means you need test tubes, and you need spatula, you need test tube holder. Okay, so the special apparatus is required, and also we need skill, a little skill is required to do this semi micro qualitative analysis. So that is why this is classified under semi micro analysis. And next, the third one is micro analysis, when extremely small quantity of sample and reagents are available for analysis. So we are going to do micro analysis. And okay, so here this analysis is carried out using milligram scale, and this is speedy and more economical compared to the other two methods of analysis. And in this method also special apparatus and skills are required. And for example, flame photometry. So flame photometry experiment is an example of micro analysis. So what is meant by flame photometry? What exactly we are going to do in flame photometry means, so in flame photometry, so we are going to determine the percentage of sodium or potassium or the many other inorganic salts so which are present in water sample or any other blood fluid samples or other samples. So we are going to determine what is the percentage of elements, that is inorganic salts, present in the given sample. Okay, so this experiment is there in your syllabus, but it was only for the demonstration experiment. But under Star College scheme, we have proposed that this flame photometry experiment will be carried out hands-on by the students. Okay, so that is why you will be doing this flame photometry experiment once the college resumes. Okay, so we have two flame photometry instruments which has been purchased under Star College scheme. So as soon as you come to the college, we'll have the experiment, and we'll see how the micro analysis is done with the help of flame photometry, that is determination of concentration of sodium or potassium is carried out with the help of this. Okay, so then it will be much more clear. And the fourth type of qualitative analysis what we do based on the sample size is ultra micro analysis. Okay, so this technique is used when extremely small quantities, compared to all the three other qualitative analysis, when we have extremely small quantities of sample and reagents, so then we will employ ultra micro analysis. Okay, so this method requires special instruments and specialized skills. For example, spectrophotometric analysis. Okay, So this spectrophotometry is a very beautiful instrument which is used to find the concentration and also the intensity of the light, which records based on the color. So many of the applications in the field of chemistry, in the field of medicinal chemistry, that especially uses spectrophotometric analysis. So this also we will do the hands-on experiment once you come to the college. We have purchased 2 spectrophotometry under Star College scheme, and this will be utilized. So you students will be easily handling it. I will teach you how to use this spectrophotometric instrument to analyze various different samples. Okay, so this example comes under ultra micro analysis. Okay, so these are the 4 different types of classifications which we do depending upon the sample size or the amount of the sample available for the analysis. Okay, and the next type of classification is types of tests. Okay, so different types of tests. So the first one is preliminary or dry test. So in this test, the sample or salt is tested in a dry state. That is why the name dry state. We need not to prepare any solution. Okay, so we need not to prepare a sample solution. Directly we can take the sample which is given and it will be tested. And okay, so this test may furnish important information which simplifies the further course of analysis. Okay, so this is not the conclusive tests. Okay, so these are not conclusive, whereas these preliminary tests, they are just a hint to the experiment. So what they will do, so they will simplify, they will give some important information regarding the sample, and they will simplify us, they will give some clues, hints for the further course of analysis. Okay, so the some important clues which we get from these preliminary or dry tests, so that will be used to find out the certain radicals, and little time should be spent on these tests in the actual progress of the analysis. And okay, so some of the important dry tests what we are going to do is, so the first one is general appearance. Okay, so by doing the general appearance test, we can get a clue like whether it is an organic compound or an inorganic compound. How? Usually organic compounds are amorphous and inorganic compounds are crystalline in nature. Okay, so by seeing the general appearance, so if it is amorphous, then we can come to the conclusion that, oh, maybe the compound or the sample which has been given for the test, it is a organic compound if it is amorphous. If it is crystalline, then I can say that, oh, maybe it the given sample is inorganic. Okay, so likewise by doing a general appearance test, so I can get some clues or hints. And okay, next is about color. By looking at the color also, I can say whether it is organic compound or whether it is inorganic, or which element is present. Say for example, if copper is present, so then I will get a blue color. Okay, so if sodium is present, so then we'll see means pale green color. So like that, by looking at the color, so where I can get some clues and hints, and also the smell. Okay, so whether it is a fishy odor it is giving, or pleasant smell does it have, or pungent smell does it have. So by seeing the smell also I can get some clues and hints. And next is action of heat. So what happens when it is heated, so whether it is going to decompose or whether it is going to change state from solid to liquid or liquid to gaseous. So how the action of heat is going to work on that particular given sample, so that also will give some hints and clues. And next is flame test. So while doing the flame test, whether it burns with the sooty flame or whether it burns in a bluish flame, so by looking at that, so I can get some clues and hints. And okay, so these are some of the preliminary or dry tests what we are going to do. And the second type of test is wet test. Okay, so as the name itself says, in wet tests, first of all the sample is dissolved in a solvent. That means we have to prepare the sample solution. Here we cannot use directly the given sample to do the tests. So first the given sample has to be converted into a sample solution. Okay, so here sample is dissolved in a solvent, and this solution is used to test the presence of constituents of a sample, and then they are further confirmed by the dry tests or confirmatory tests. Okay, so the presence of constituent radicals can be tested by dry test and confirmed by wet test. Okay, so this is how a wet test is performed. For example, the presence of halides like chloride, bromide and iodide is tested by dry test using concentrated sulfuric acid and confirmed by wet test with silver nitrate. Okay, so this is one beautiful example of wet test. So what is done here? So say for example, in the sample what has been given to you, if halides are present in that is chloride, iodide or bromide is present, so how to confirm that is, so I will take the given sample in a watch glass or in a test tube, and then I am going to use concentrated sulfuric acid. That means concentrated sulfuric acid is added to that given sample, and it is confirmed further with the silver nitrate. If I add silver nitrate and if it is give milky white effervescence, so it confirms that halides are present in the given compound. Okay, so this is how we are going to do the wet test. And the next one is the basic radical of inorganic salt is tested with the group reagent after preparing original solution of salt in water or in dilute hydrochloric acid. Okay, so these are the two examples of wet tests. And the third type of test what we are going to do under qualitative analysis is spot test. So the spot tests are carried out by bringing together one drop of test solution and one drop of the reagent on a solid surface like porcelain plate or a glass plate or a paper. Okay, so that is why the name spot test. Okay, so here spot is used, that is one drop of test solution and one drop of a reagent is used. So since we are using drops, that too those drops are spotted on a solid surface like porcelain plate, glass plate or a paper, so hence we call it as spot test. Okay, so the spot tests are usually applied to micro and semi micro techniques for identifying ions or compounds. These tests can be carried out quickly and economically. Okay, so these tests are carried out quickly and economically because just one drop of sample solution and one drop of reagent is required, and it is very quick. We can see the reaction, we can see the changes what has been taking place through our eyes, and we can come to the conclusion. Say for example, so ferric ion, that is the presence of ferric ion, can be tested by placing one drop of the sample. Okay, say for example a sample has been given to me and I have prepared the sample solution, okay, in which it is soluble. By knowing the solubility I have prepared the solution, and one drop of sample solution I have spotted on a porcelain plate. To that what I am doing is I am spotting one drop of reagent, that is ferroin. Okay, so I hope you all have seen ferroin indicator. So ferroin is an indicator which has a pink color or pale red color. Okay, so the IUPAC name of ferroin indicator is 7-iodo. So this is 7-iodo, 8-hydroxy quinoline 5 sulfonic acid. So this is the structure of ferroin, and when we add one drop of reagent, that is ferroin, to the given sample solution, so if I see green color, if the formation of green color is observed, then it confirms the presence of ferric ion. Okay, the sample solution what I have prepared, that will be colorless, and the reagent what I am using, that is ferroin, it will be a pink color or a pale red color. Okay, and if I observe the formation of green color, then it confirms the presence of ferric ion. Okay, so this is how we are going to do the spot test, Sant. So these are the three types of different tests what we are going to do in qualitative analysis. Okay, so there I will stop. So in our next class we are supposed to discuss about quantitative analysis and what are the different methods of quantitative analysis, and we'll see one by one what is meant by volumetric, gravimetric and instrumental analysis under quantitative analysis. Okay, so I will stop here and we will meet in our next class. Okay, thank you dear students.